Welcome once again, my dear students. This is Meiosis Senior Lecturer of Master College. Today, I will discuss some topics from Chapter 2. The name of the chapter is Cell Division. Today, my topics are steps of Meiosis II cell division, importance of Meiosis cell division, differences in between mitosis and meiosis, crossing over, and the significance of cross-difference. Now, I will discuss the steps of Meiosis II. You know, meiosis cell division is divided into two phases. One is meiosis 1 and another one is meiosis 2. Meiosis 2 is again divided into four substeps or subphases, that is known as prophase 2, metaphase 2, anaphase 2 and telophase 2. It is quite similar to metaphase cell division, but it occurs in the haploid nuclei. You can see after the meiosis 1 cell division, the mother nucleus, it divides into two daughter nuclei, and the number of chromosome in case of meiosis 2 or meiosis cell division, it becomes half that of the mother cell, and it is a continuous process as like as the mitosis cell division, but sometimes it takes a little bit time after the telophase 1 to start the meiosis 2 cell division, and this gap in between meiosis 1 and meiosis 2 is known as interkinesis. Now, first of all, prophase 2. You can see I use here two colors. One is green, another one is red. It indicates that homologous chromosome, the parental cells, that is one pair comes from the father and another pair comes from the mother, and already due to crossing over, different pairs of the chromosome already has been exchanged. Now, what happens during prophase 2? During this step, the chromosome become shorter and thicker due to the dehydration of nuclei. Then at this stage, you can see that the chromosome can be observed under a compound microscope. At the end of this stage, and the nuclear membrane and nucleolus disappears. And during this prophase 2, the chromosome become divided into two chromatids. Now, metaphase 2. What happens, or what are the characteristics of metaphase 2? In case of metaphase 2, as like as the mitosis solution, you can see that spindle apparatus are produced here also. So spindle apparatus that are produced during metaphase 2, and at this stage the chromosome becomes shortest and thickest, and you can see the centromere of the chromosome, they remain attached with the spindle apparatus. These spindle apparatus or fibers are known as traction fiber. So during the metaphase stage, the spindle apparatus with two opposite poles or two poles are produced. The centromere of the chromosome remain attached with the spindle apparatus or traction fiber, and the chromosome or chromatids, they remain at the equatorial plane. So these are all about the metaphase. Now, anaphase 2. During anaphase 2, the chromosome divide into two daughter chromatids, and at first the centromere divides and it pulls apart the two daughter chromatids from each other towards two opposite poles. It is happening in both cases, and during this time, the daughter chromatids or daughter chromosomes or the chromatids looks like I, J, L or V-shaped. So this is all about the anaphase stage. Next, you can draw a spindle apparatus just in between these two daughter chromatids. Okay. Next, the last stage of meiosis 2 cell division, telophase 2. You can see that during this telophase stage, the nuclear membrane again reappears. That means the new nuclei are formed, or nuclear formed. There are 4 nuclei, 1, 2, 3, 4. This is the nucleolus. So in case of telophase 2, here 4 nuclei are produced, which are not similar to the parental or the mother nuclei, because in case of the mother nuclei there were the number of chromosome in diploid state, but you can see here the number of chromosome is half. Okay, it is because, and they are also dissimilar in their characteristics due to the crossing over that occurs in prophase 1. After this telophase 2, the cytokinesis occurs, and these 2 phases, meiosis 1 and meiosis 2, after that it helps in the production of 4 haploid cell from a diploid mother cell. So my dear students, this is all about the steps of meiosis II cell division. What is mitosis cell division? You know that the process of cell division in which the nucleus and the chromosome of a cell divides only once to produce 2 daughter cells, where the number of chromosomes will be same as the parental cell, is known as mitosis cell division. It is also known as equational cell division. On the other hand, the process of cell division in which the number of the nucleus divides twice but the chromosome divides only once, for which the number of chromosomes become half in the daughter cells, where 4 daughter cells are produced, is known as meiosis cell division. It is also known as reductional cell division. Now come to the point. The differences between mitosis and meiosis you can see on the board. That is, first of all, the topics mitosis and meiosis cell division. Place of occurrence. You know that the mitosis cell division takes place in the vegetative or somatic cell, whereas the meiosis cell division takes place in the reproductive mother cell. Next, nature of the cell. In which type of cell the cell division occurs? It occurs both in haploid and diploid cell, but meiosis occurs only in diploid cell. Number of daughter cells. 2 daughter cells are produced after meiosis cell division, and after mitosis cell division, and after meiosis cell division 4 daughter cells are produced. Next, number of chromosome in the daughter cells. You know that after the division of mitosis cell division, the number of chromosome in the daughter cell will be similar to the parent cell. On the other hand, the number of chromosome will be half in the daughter cell than that of the parent cell in meiosis cell division. Now, next point. Nature of the daughter cells. Daughter cells are similar to the parent cells genetically in case of mitosis cell division. But in case of meiosis cell division, daughter cells have differences, genetic differences from the parent cell. Next, crossing over. There is no crossing over in mitosis cell division. Crossing over takes place in meiosis cell division during the or in between the non-sister chromatids, which occurs during the prophase 1 cell division, steps of prophase 1 cell division. Okay. Finally, the function. The function of mitosis cell division, it helps to grow the body of the organism. That means to help in growth and development of the body, to heal up or heal wounds. Okay. Next, the function of meiosis cell division. It helps to produce spore, gametes, and it also helps to maintain the chromosome number constant in a particular species. Now, the importance of meiosis cell division. In case of meiosis, the importance of meiosis cell division, first point is production of gametes. Due to meiosis cell division, the male and female gametes are produced, and the male and female gametes are haploid, which helps in sexual reproduction in different organisms. Point number 2, assortment of chromosome. In meiosis cell division, the paternal and the maternal chromosome assort independently, which helps in controlling the traits. Restoring of chromosome number. Due to meiosis cell division, the diploid cell, when produce 4 daughter cells, they turn into haploid cells. Ultimately, it forms the haploid gametes. And when these gametes unite together, they form the zygote, which again turns into diploid stage. Thus, the restoring of chromosome number is maintained in a particular species. Combination of genetic material. Due to meiosis cell division, the crossing over takes place in case of prophase 1 of meiosis 1 cell division. And due to the crossing over, different parts of chromosome or chromatids are exchanged in between themselves, and it occurs in between the non-sister chromatids. And due to this reason, the combination of different characteristics occurs, and thus the combination of genetic material occurs due to meiosis cell division. Reduction in chromosome number. As I have told you, due to meiosis cell division, the number of chromosome reduces in the daughter cell from the mother cell. So the number of chromosome reduces in the daughter cells. Though it is reducing, it also helps in maintaining the chromosome number constant. First of all, the diploid cell turns into haploid condition. Again, when they fuse together, they again turns into a diploid state. Thus, the reduction of chromosome number helps in maintaining the stability of a species. And production of variation. In case of point number 4, combination of genetic material which occurs, that results the production of variation. Due to the production of due to the result of meiosis cell division, it produces 4 daughter cells. In each of the daughter cells, they are different from each other, hence different parts are combined together, or they are broken from each other and they form a new combination. That is why a variation is found in those 4 nuclei which are formed after the meiosis cell division, and that helps in producing a new variation, or variation is found after that. So my dear students, these all are the importance or significance of meiosis cell division. Now the last topic, the mechanism of crossing over. You know, during zygotic stage of prophase 1 of meiosis 1 cell division, the homologous chromosome forms the pairs, this which is known as synapses, and each of the pair is known as bivalent. So this is the bivalent. In case of pachytene stage, each of the chromosome divides and form 1, 2, 3, 4 chromatids in each bivalent, and these are known as tetrad. At the end of this pachytene stage, the non-sister, in between these non-sister chromatids, they form a X-marked part or X-like structure. We can see this X-like structure. This is known as chiasma. And at the point of chiasma, the non-homologous chromosome, or the in between two non-sister chromatids, they form a loop. And these actually at the point of chiasma, in between two non-sister chromatids, they exchange their parts due to continuous or extreme dispersion or repulsion force. And what happened next? So at the end of the pachytene stage, you can see the two non-sister chromatids, they exchange their parts, and there is a new combination in these 2 chromosomes, or in these 2 parental chromosomes, and 2 enzyme. One is known as endonuclease enzyme that helps to break these 2 parts of chromatids, and another enzyme known as ligase enzyme that helps to rejoin these 2 parts. So thus the crossing over takes place in during the pachytene stage. Now the significance of crossing over. What are the significance? As you can see that during this crossing over, the different parts of chromosome are exchanging in between themselves. That is why new traits are found in the gametes. Not only that, different we can determine the genetic mapping. We can insert the desired DNA. which plays an important role in genetics. So these are the significance of crossing over. So my dear students, this is all for today. From my today's lecture, I think you will be able to explain the different steps of meiosis II cell division, then the importance of meiosis cell division, the difference between mitosis and meiosis cell division, steps of crossing over or the method of crossing over, and the significance of crossing over. In my next class, I will be back with a new lesson. Till then, take care.